The next speaker now is Josie McGovern, and Josie has been waiting there patiently. She is she did a postgrad diploma of science with me, and then it now is doing a master's. And she is studying the fungi that are in the dung of takahe, and I'm going to just leave it to her to tell you all about it.
Good afternoon, everyone. I'm Josie and I'm a master's student at the Otago Botany Department. And I'm supervised by David, Tina and Andy. I've really enjoyed listening to everyone's talks today, so good job on that. And today I'm going to be giving a talk on takahe and how they interact with fungi. So to do this, I have been studying dung. And why would anyone want to look at dung? Well, you can determine a wide range of biological information about an animal from its dung, and most importantly, in a non-invasive way. And this can range from anything about diet to health and disease. And a lot of study has gone into coprolites or ancient fossilized dung, and this helps us to understand how an organism might have interacted with its environment at the time of its existence. But I am interested in what dung can reveal about fungi. So fungi follow many different lifestyles, as we all know. Saprophytic fungi have the ability to break down the lignocellulose matrix, which is something which other organisms often struggle to do. And one such group of these are coprophilous fungi, also known as dung fungi. And they help to recycle nutrients and convert carbon into a more appropriate form for soil uptake. And they have to survive the digestive process of whatever animal consumes them. And it's thought that this process is actually a requirement for a lot of them to germinate. In New Zealand, we don't have too much literature on coprophilous fungi, but we do have Anne Bell's beautiful illustrated guide that was produced in the 80s. But since then, we've had quite a DNA sequencing boom, and so we have the ability to add a lot more to that. So in 2020, as part of my postgraduate diploma, I wanted to investigate coprophilous fungi and how they might compare between animals living in contrasting habitats. And for this, I settled on two different grazing animals, the takahe, as I mentioned before, and our domesticated sheep. So sheep are grazers. They're distributed nationwide, and so that makes it easy for me to get my hands on their dung. They've got great cultural and economic importance to our country, and for anything to survive out the other end of them, it has to pass through 4 stomachs. So I collected, and others collected for me, 50 sheep dung samples from 5 different farms. 2 of these were local in Dunedin, and 3 of them are from neighbouring farms in Rotorua. And our other animal, the takahe, who is rare and endemic to New Zealand and can be referred to as a zombie species because they were thought to have been extinct for about 50 years, only to be rediscovered by George Orwell in 1948, where they have a wild population in the Murchison Mountains. Takahe have an extremely heavy grazing behaviour, and they like to eat tussock tillers. They spend 90% of their daytime foraging, and they also follow a seasonal migratory pattern where they move out of the tussock land into nearby beech forests in wintertime. So captive takahe defecate on average 45 times during 14.5 hours of their feeding activity, and it's estimated that wild populations would do this 4 to 5 times higher per hour because they need to get a bit more nutrients out of what they're getting, and they're not getting supplementary feed like they would in captivity. So pretty much, Takahe exhibit such a high interactivity with their environment that they have the potential to come into contact with a great number of fungi. In 2007, unfortunately, the wild population was halved by stoats following a beach masting event. And as of October last year, the population consisted of about 440 individuals. And this includes those that live in sanctuaries. And in sanctuaries, they are able to live up to 22 years old. And they're safe there because they're not coming into contact with animals and stoats and things like that, except for humans. So in Dunedin, we have the beautiful Orokonui Eco Sanctuary, and it's about 20 minutes drive from town. It's 307 hectares of forest surrounded by a predator fence. And in Orokonui, they have 2 breeding pairs of takahe, and I've circled on this image 2 different sites where the takahe pairs live. Site 1 here is a wetland area surrounded by eucalyptus regnans, and site 2 is more of a native low forest, and there is some kānuka nearby. And so I collected 20 samples of dung, 10 from each of these sites, and added with sheep dung samples, that makes 70. The DNA was then extracted using a power soil kit, and ITS2 regions were sequenced. And this gave us back 2,041 ASVs, which were then analysed using RStudio, and any identities were assigned using the Unite database. So here you can simply see that takahe have quite a lot, are significantly more rich in species, fungal species, than in the sheep, which is quite interesting and is probably related to the diversity of plant species and all other species that they have access to compared to the sheep who are grazing in a paddock. And here is an ordination plot which demonstrates the fungal community dissimilarities between sheep and takahe dung samples and the different locations that they're from. And in this red oval here, you can see that the bulk of the sheep samples all have clustered here. And it's quite interesting to see that a particular Dunedin farm is more similar to a farm from Rotorua compared to another farm within the same region. And here, forming an L shape, are the takahe dung samples. The sample that goes up the left side here is the dung collected from the eucalyptus site, and the darker blue ones were from the low forest site. And it's quite interesting to see how different those 2 sites were from each other and from the sheep. So there are many routes of fungal dispersal, and the most obvious route would be wind, and many but not all fruiting bodies are designed to help promote this. The caps are raised, holding spores up on the stipe, creation of airflow by evaporating water, catapulting, such as with Pilobolus. But how much distance does this actually afford fungal spores to travel? And I think it suggested that they travel around up to 1 metre squared. So how could they potentially move further than that? And biotic factors offer interesting alternative routes of dispersal. And there's been lots of documentation about mammals and marsupials interacting with fungi over the world, and as well as insects and lizards. But in New Zealand, historically, we don't have any other mammals except for the bat, and so potentially they have been interacting. But we do have, or have had, a lot of birds. So do avians spread spores? And the study that Amy mentioned at the beginning of the day is popping up again. And it's basically a study where in Patagonia, some birds were investigated as to how they may have been spreading spores for truffles and other mycorrhizal fungi. And Patagonian forests are dominated by Nothofagus, similarly to New Zealand. And this study sequenced dung and soil samples, had visual confirmation of mycophagy occurring, which is exciting, and high amounts of ectomycorrhizal fungi were detected in both of the dung from the birds. And a high frequency of this were quaternarious sequences. So ultimately, they have decided that these two birds play an important dispersal role for the forests of Patagonia, contributing to forest diversity, health, and movement. But what about in New Zealand? So it's still up for debate whether New Zealand birds engaged or still engage in mycophagy. And we know that our birds are attracted to brightly and white coloured fruits and berries over those which are more dull, green and brown. And many of these colours, as mentioned many times, are represented in our fungi. We also know that moa purposefully ate large quantities of small white stones or gizzard stones to help them with digestion. And potentially small white fungal fruiting bodies could be consumed like this as well. So we do know that fungal DNA is present in moa and kakapo coprolites, and it's been suggested that they might have helped spread New Zealand tree lines by dispersing mycorrhizal fungi. But it's quite hard to know if they were deliberately engaging in mycophagy or if it was just by mistake. So we have lost around 41% of our Indigenous species and used to have 31 flightless species. And all of these ground-dwelling birds would have had high contact with fungi in this space, so it's difficult to know what their role in forest dynamics might have been. Today we have 15 flightless species remaining, and this of course includes the takahe. And so takahe provide a great opportunity to gain insight into how these missing birds might have influenced their environment. So, as well as DNA sequencing the dung, I also did an experiment where I plated out dilutions of dung on agar, and I was able to isolate pure cultures. And this proves that fungi are indeed viable after being excreted by the takahe, and that this was not only coprophilous fungi. So you can see a few pictures here. The top three were isolated from sheep dung, and the two on the side were isolated from takahe, and they all have various roles in the environment. And down the bottom there's a popular edible variety, Flammulina veludipes. So I think this is quite exciting to contribute to my research. And what began as a study focused on coprophilous fungi very much expanded, and DNA sequencing opened my eyes into what else was going on in the dung. And so, were there any fungal species of import to New Zealand forests present in my takahe dung? And this is yes. Quaternarious sequences were identified, and these were only found in the takahe dung, none in the sheep. And this is an ectomycorrhizal genus with many species known for its crucial associations with New Zealand beech and manuka and carnuka. And the takahe dung revealed 13 different species of quaternarious, which compared to only 7 species recorded from collection visits to Orokonui by the department. Of course, this has changed over the last couple of years as more collection trips have happened, but I think that's quite exciting that just in one day of collecting dung, we were able to see what else had been living in Orokonui that we hadn't previously seen. And it so happened that in a couple of the dung samples, Cortinarius cooler was found at much higher reads, and this might potentially suggest that there was presence of mushroom flesh in the dung. But who knows if the takahe are engaging in mycophagy or not. So I'm still researching at the moment, and I have undertaken 6 more dung collection trips to Orokonui over several months, and each time I collected 30 samples, but this time 15 from each site, and this totals up to 180. So in comparison to my first only 20 samples, hopefully I am going to get a lot more data to look at. And I want to know what fungi do these contain, any of these important mycorrhizal fungi, and how might the fungal species present change or fluctuate over the time that they were collected, and what insight does this give us into the ecosystem services of ground-dwelling birds and fungal dispersal. So to accompany this, I ran a glasshouse experiment where I grew seedlings of manuka and Eucalyptus regnans, and I inoculated them with a lovely takahe dung slurry and grew the seedlings up, and I've since harvested the roots. And the next step for this is to do some root staining, and hopefully we might see that the roots have formed some mycorrhizal associations. And this will be hopefully supplemented as well with some DNA extractions. So all in all, some important learnings from my research are that takahe dung has great fungal richness. Fungi from multiple functional groups are viable post-excretion, and therefore takahe can offer another avenue of dispersal for fungi. DNA sequences from important mycorrhizal fungi have been found in takahe dung, and there is a potential that takahe may be involved in the dispersal of mycorrhizal spores. DNA analysis of dung from certain species offers an extremely valuable tool to gain insight into the fungal realm. So I'd just like to thank a few people for their help along the way, especially my supervisors, and of course the animals who donated to the cause. And thank you for listening. I hope you enjoyed my talk.
Great. Thank you, Josie. Okay, getting a clap there, that's good. You can stop sharing your screen if you like. I know we're running late, but has anyone got maybe just a question or two we can squeeze in? See, Teresa said she's had to go to another meeting. Oh, David White. Okay, let's there you go. You should be able to unmute yourself now, David.
That was fascinating. A quick question. The farms that the sheep came from, were they beautiful grass pastures like I see some sheep farms, or were they manuka scrubby sheep farms, which also exist?
I believe they're all nice grassy paddocks, including my own home farm. So I'm very well acquainted with the sheep and where they live. Yeah, all nice grass pastures.
Could be interesting to see the sheep dung from the manuka-y scrubby kind of farms to see if they've got an increase in things. But a fascinating research. Thank you.
Great. Thank you. Nicola and Rosalind have just said great, so that's awesome. Thanks. Okay, that it? Looks like it. It's getting late. Thank you, Josie. It is an interesting project, pretty exciting.